All right. So thanks everyone for tuning in or sticking on after the first presentation today. My name is Chanel Hayson. I'm the Director of Outreach and Community Relations for the nonprofit Elakha Alliance, and we are about to start our second presentation of the day. So let's see. Hold on, Matt. Let me just work this presentation real quick, this last slide. So if you're unfamiliar, the Elakha Alliance is a nonprofit here in Oregon. Our mission is to make Oregon's marine and coastal ecosystems and communities more robust and resilient by restoring a healthy population of sea otters back to the Oregon coast. And our next speaker is Dr. Matt Edwards. He's a professor of biology at San Diego State University, and he focuses on patterns and variation of giant kelp forests at multiple spatial and temporal scales throughout the species' geographic range in the Northeast Pacific Ocean. And today he'll be focusing on one of the chapters of the Sea Otter Conservation II book, Sea Otters and Rocky Reef Communities. So now I will pass it off to Matt.
Thank you so much. First of all, I want to thank you very much for inviting me to do this. It's a great opportunity. I do look forward to meeting many of you in person, hopefully next year. I am planning a move actually to the Oregon coast sometime in the late summer, fall, where my wife and I are moving to Waldport, actually, where we're building a house. So I'll be in the area, and hopefully I'd like to continue a collaboration with many of you in person. Today, I want to talk to you about one of the chapters we have in this book. Let me do a share here. Okay, and 1 second. All right, I'm going to 1 second here as I navigate the share buttons.
It's always fun.
It's always great. Yes, I know. It's let me make sure I'm on the right one. You get so many things opened up. And this one right here, I think, is the one I want to go with. Okay. All right. I think this is good. All right. As I said, thank you very much. So in the introduction, it was nice. You were told that I do focus on giant kelp forests, primarily along California and Mexico coastlines, but I spent quite a bit of time up in the Pacific Northwest and in Alaska. I'm studying the sea otter populations and their effect on the kelp forests up there. So today, what I want to do is talk about sea otter communities and rocky reef communities. And in essence, I kind of want to follow up on some of the things that Jim kind of alluded to on his very last chapter he talked about, where it was the larger consequences of sea otter loss and recovery on the ecosystem. So what I want to do is to focus on some ecosystem services. My co-authors on this chapter were Jane Watson and Brenda Konar. And obviously, we dedicated this to Jim Estes, who were all three of ours PhD advisor. So I was invited to go up to the Aleutian Islands in my early graduate days, actually my mid-graduate days when I was beginning my PhD at UC Santa Cruz. And I spent a bunch of time up there. And after I got my faculty job, Brenda and I in particular continued for many more seasons up there studying kind of the broad scale effects of the sea otter loss and the phase changes we saw. So that's what I'm going to focus on today. And I'm going to start this by just saying what thing I believe is one simple truth is that some species have larger impacts on their ecosystems than others. And so in this case, sometimes we're talking about the sea otters. I'm going to talk about the kelp. Many species are foundation species. They have large disproportionate effects on their communities. They provide critical habitat and energy. They provide structure. And we can see this across a broad range of different ecosystems. And if you think about just a forested community, right? You walk into a forest, it's a very different environment than right outside the forest. It's cooler, it's often more damper, those lower light levels. There's a whole different species assemblage that's within the forest. And sometimes we lose the forest, and we can see these in patch gaps where suddenly the ecosystem changes. It's brighter, it's drier, and we can see differences in the species assemblages there. And so Trees are often considered foundation species, right? They modify the microclimates, they regulate primary production, they change energy flow and nutrient cycling, they enhance biodiversity, and loss of them or deforestation can lead to loss of these ecosystem functions. And so I put this juxtaposed next to a kelp forest picture, and I want us to think about kelps doing the same thing, right? And so kelps as foundation species is a whole broad range of different species of kelps throughout the world, generally from temperate to polar waters. And they're perfect examples of this. And so one of the things just to start with in a review by Dan Reed and Mark Brzezinski, looking at the effects of kelp forests on just global carbon cycling and standing crops. And on the left of this, you see on the area, these white bars are these different kinds of forests, tropical rainforest, seasonal forests, boreal, evergreen, deciduous forests. And you can see there's the standing crop of kelp upwards between 1 and 2 kilograms per meter squared. And if you look on the right side in the yellow bars, these are three different kelp forest species, Laminaria, Aclonia, and Macrocystis. And they're on par with those, maybe a little less as far as a standing crop, but they hold just about as much carbon as as terrestrial forests. But if we look at net primary production and the amount of carbon fixed per unit area per year, we actually see that kelps exceed typical forests. And they're turning over much rapidly, but they have high rates of primary production. And in some work we did with Chris Wilmers and Jim and Christian Leandri and Brenda Konar, we looked at throughout the Sea Otter range, and we looked at the amount of kelp within the standing crop, amount of carbon within the standing stock of different kelp species, and what was lost when we lost these kelps. And we could do this by collecting the kelps, drying them, measuring the carbon in their tissues, and then modeling the amount of available substrate and how much kelp has been lost. The take home from this is if we just look at the carbon density, about 0.18 kilograms per meter squared, we're right on about that mark we saw in Breen Brzezinski. But the scenario without the kelp is we lose about 99% of that ability to hold on and store carbon in a standing crop. Now, this is not permanent carbon sequestration because kelps turn over, but it is carbon locked up within the ecosystem at any one time. And so what we call the kelp effect is just about the entirety of what's there. And so when we think about kelps, they're distributed worldwide, generally not tropical, they're anti-tropical, or there are some kelps observed near the tropics, particularly around the Galapagos Islands. And they get up to about 6 degrees latitude in the south along the coast of Peru where there's strong upwelling. But the take home from this is we see kelps around the world, but there's a whole number of species of kelps. I'm going to focus up here in the Pacific Northwest, particularly around Alaska. I'll touch on the Gulf of Alaska and California at the very end of my talk. And so I like to think about kelps when I start thinking about who was the first kelp ecologist. We have to go back to the voyage of the Beagle, right back in the 1800s. And as Darwin took the Beagle down around the tip of South America and up the South American coast, he made some observations. And I love this quote from the voyage of the Beagle, where he says, "The number of living creatures of all orders whose existence intimately depends on the kelp is wonderful. I can only compare to these great aquatic forests in the southern hemisphere with the terrestrial ones of the intertropical regions. Yet, if the latter should be destroyed in any country, I do not believe so many species of animals would perish as under similar circumstances as what happened with the kelp." So he's basically saying, compare these to the great tropical rainforests, the kelps are right on par. So the question then becomes, and where I'm going to focus most of my talk today, is what happens if we lose kelp forests? And in California, this is a giant kelp forest in Monterey. But when we lose the kelp forests in California, we often see them piled up on the beach like this. I love that little bit that Jim presented on the energy transport onto the shore. This is an energy subsidy for the sandy shores or the intertidal. But what happens to the subtidal forest when we lose these things? And so the example I'm going to talk about are sea otters, kelp forests, and coastal food webs. We'll start there. And this is again in collaboration with Jim Esses and Brenda Konar, this first set of talks. And this is going to focus mosely on the Aleutian Islands, the archipelago that extends west from the Gulf of Alaska out toward Russia. And sea otters are the kind of the main player of the day. These are sea otters wrapped up in giant kelp, not the kelp in the Pacific Northwest, but it's a nice picture of one. And it kind of starts off with the story that many of us are aware of that Jim postulated in the late 1990s with killer whales and a dietary renaissance or a changing of the way killer whales fed. Typically, we thought of them feeding offshore on great whales. During this time, we started seeing them in our dive sites, and this is a killer whale in the Aleutians with kelp draped off its dorsal fin. So there was some sort of a change in their feeding behavior that went on. Um, and what we saw with that co-occur with this is we went from the status of having kelp forests. This is a Eularia fistulosa or a dragon kelp forest with a number of species of Saccharina, laminaria down below. It went through a transition period as urchin populations began to explode. These are green urchins on the benthos, and we've lost all the benthic algae, and we have just some Eularia kelps on the tops of pinnacles where wave beat back and forth, and this whiplash of their reproductive sporophylls, these blades at the bottom, tended to keep the urchins at bay. But ultimately, we ended up in urchin barren grounds widespread throughout the Aleutians, and often with really, really high abundances of sea urchins. So, what some of the work that Jim did is not only document this, but would weigh pieces of kelp, and he and his colleagues would place these things on the benthos, tie them to metal bars so they'd stay on the bottom, put them out in the urchin barrens, and then these urchins would just swarm it and start to devour the kelp. So this gave the ability to characterize the changes in several ways. And this is an image from SD's seminal paper in 1998 in Science, where we see sea otter abundance historically going back to the 70s and 80s, was high, began to decline throughout the 80s and into the 90s, and it's dropped considerably further. Urchin biomass went from very low, exploded. Grazing intensity, as measured by the percent loss of the kelp on those bars, exploded, and the total kelp density crashed. So this trophic cascade, which started with this change in the diets of the killer whales, played out such that I'm going to focus my story on today on what happened with this huge increase in sea urchins and this absolute large loss of kelp and fleshy algae. So one of the things that we did, Brenda and I wrote a NSF proposal, which was funded, luckily, and was to ask the question, what happened with this transition? And this is following some other work by Estes and his colleagues, which we can look at the rock greenling abundance, and anywhere you see there's an orange bar, consider that a sea urchin dominated, no kelp, no otters. And going back from the 80s to where and on Attu and Shemya, this is before we saw some of the declines in some of the more eastern islands, but out in the west, we see that fish abundance was low going from the 80s to the 2000, and same thing at Shemya. But at Amchitka and Adak, as you moved east, before these huge declines, we saw large fish abundances in these sites, and they crashed following this trophic cascade and the loss of the kelp here. So we saw changes to the fish abundances. Work with Dave Duggins, we saw as the kelps were there and they began to break down and create particulate organic matter, right? Barnacle sizes decreased, mussel growth rates decreased. We saw the changes in, say, glaucous wing gull diets. Where they used to eat fish, as those fish abundances disappeared, they weren't eating fish anymore. They started eating more sea urchins and other invertebrates. And all the way up with looking at Anthony's work, we saw that all the way up to kind of bald eagle diets. We saw, again, a change where they used to eat mammals, a lot of sea otter pups. That changed. Fish abundances went down, but bird abundances went up. And these are primarily eider ducks, which were in competition with the sea urchins, with the sea otters, and their populations began to increase as urchins became more abundant. So this changed a full of the food web, And so following on these changes, which again were not mine, they were documented mostly before I started working with Jim. But following this, Brenda Konar and I were lucky enough to have several NSF grants to go back to the Aleutians. We were able to take the RV Tommy Thompson out of Washington, the Point Sur, which used to be out of Moss Landing Marine Labs, and then the RV Oceanus several times out of Oregon State. And we went up to the Aleutians to document what were some of the losses of ecosystem function. And so, for those of you you may have be familiar with the area, the Aleutians are a beautiful place to work. These nice, iconic volcanic mountains coming right out of the ocean. Sometimes you look out the porthole of the window. It's very often foggy up there. You might see days like this. Other times you look out the porthole and you see days like this. And I like this image here because if I can get this video to play, it doesn't look like it's going to. During these times, we would have huge waves coming over, and this is a video of these waves washing over the side and completely inundating the side with water. And we would go out in small boats in these times and do our dives. And this is one of these days that's a little rough that's to be out there. Now, get this thing loaded. But other days, the Aleutians could be beautiful. This is Island of the Four Mountains. You can see, I believe this is the RV Point Sur, which is about a 135-foot boat. And we were able to hike up and look down on it. When you had days like this, perfect days to work up there. It's beautiful diving throughout the Aleutians. And so we went out to the first thing to look at changes in biodiversity. And so we looked at the percent bottom cover of different organisms. We did quadrat scrapes, which we collected everything within these quadrats, brought them back to the ship, weighed them, and we got all the epifaunal taxa, the things that lived on top of the rocks, except those things that were very solidly attached. Some of these things were hard to get off. And then we did visual swaths where we counted large mobile invertebrates, and we estimated fish and the kelp canopy counts. And so this is just a picture of working up there, doing sea otter counts within sea urchin counts within quadrats. We would then place quadrats down to haphazardly placed areas, collect everything that was in the quadrat, bring it back to the ship. And this is a collection of sea urchins to get a size frequency distribution, which I'll show you in a little bit later. Then back on the boats, we would weigh everything, divide them into taxa, and get an assessment of the whole biodiversity and who was there. And the take home from this, as we went from the kelp forest through those transition habitats where we lost all the bottom seaweeds, but we had the kelps just on top of the rocks into the urchin barrens. And what we see, we can look at kelps, different red algae, other brown algae, green algae. Really, it's mostly about the kelps that were lost as far as the biomass goes. So the story I'm going to focus on is really about the kelps. Invertebrate abundances, we can see the same thing going from the green, which is the kelp forest, through the transition into the blue, which are the barrens. These are Strongylus and Trotus. These are the green urchins. And what we see is it's mostly about the urchins. So as I said before, these are the two things I'm going to focus on mostly. This is an MDS plot, and for those of you who have seen these before, you know the layout. Essentially, these are all the different samples we've taken in our kelp beds, our transition, and our barrens across the Aleutian archipelago, and points that are closer together are more similar in species assemblages, and points that are further apart are more dissimilar. And what we see is the kelps kind of group all the way over here on the left, and the urchin barrens and the transitions group on the right, showing that the kelps were different from the urchin barrens in the transition. And these vectors allow us to overlay on these to tell us who's really driving this. And what we really see is we see Eularia and Saccharina. These are two kelps that have their vectors extending to the left, meaning that if you're on the left side of things, you have more dragon kelp and more benthic kelps. And if you're on the right of things, you have more sea urchins. So we see this play out nicely in the community where the transition zones and the urchin barrens have more urchins, and the kelp forests have more kelp. Not super surprising there. And if we were to kind of summarize everything, looking at the biomass here, the kelp forests have canopy kelps, subcanopy algae, and the sea urchins are sparse. But the transition have those canopy kelps up on top of those rocks, but they don't have the subcanopy algae, but they have lots of urchins, and the urchin barrens algae are essentially absent, and the sea urchins are really abundant. So that's kind of the way I've set it up. They match it nicely. So the conclusion kind of from setting up the story, which is going to get me into ecosystem function, is that the loss of the kelp forest, that is deforestation, resulted in widespread changes to the benthic diversity. Algae and most invertebrates were more abundant in the kelp forest, while urchins were much more abundant in the barren grounds and the transition habitats. So what about the fishes? To do this, we worked with a graduate student, Terrell Effred, from Brenda's lab, and that's a little a makeshift kelp forest here in the yellow, and that's land. That's a pretend zodiac. The zodiac would come up to the site, encircle it with a GPS, convert this to GPS waypoints, and then so we can map the kelp forests. We can get a size of them and their size. And then we would do a transect on the edge of the forest, a transect in the middle of the forest. And this is a nice little video that Terry made where the two divers would go. One diver would survey the benthos, one would do the midwater, and we'd do these long 50-meter transects, and you'd swim a box 2 meters by 2 meters wide to get an estimate of the fish density. And then all fish were identified and counted. So the divers would go along. Swimming the kelp forest would generally make the fish kind of disperse. You'd reel up one of the tapes, the fish would go away, and then the divers would continue along the bottom, transect, and they would essentially look at the rugosity, the topography of the bottom using a bar and chain. They'd do the understory abundance and percent cover estimates of within quadrats of all the cryptic species. And so they'd go along, measure the rugosity, and then measure the cryptic species. And then they would come back once again with flashlights, swim along, look at every crack and crevice, and get the things that were hiding. So it gave us a really nice estimate of what fishes were there. And we did this at 30 sites at 15 of the islands throughout the chain across a 1,500-kilometer range. And of this, we really were able to estimate the abundance and the story for 25 fish species. The fish parameters that were measured are the species composition and the fish abundances. And the habitat parameters we measured were the caniform kelps, which there are two of them, Eularia, the dragon kelp, mostly toward the west. And on the east side of the Estamilgo Pass, we got the bull kelp, Nearshore, which is the one you are familiar with along the Oregon coast. There are 6 species of understory kelps, 5 different substrate covers, understory algae, 5 species, encrusting invertebrates. We had the kelp forest size, the depth, the rugosity, bottom type. We got measures of salinity and temperature. So we tried to characterize the entire habitat. The reason why we did this was to understand what's happening with the fishes, and if they changed, what are they now associating with? And so the first thing we can look at, these are all the species across this colored bar. And as we look, there seems to be the very east side of the Aleutian and the west side look different from one another. And of the 25 taxa we observed, really 4 taxa contribute to 55% of the variation among these big regions. These were our rockfish, our cods, our greenleaves, and our sculpins. And so we are surprising. We looked at this. We said, okay, let's look at the abundance of these things between the areas where kelps have been lost and where kelps were still there. And it was really surprising to see that there weren't any significant differences between the two habitats. That is, the kelps were lost, but we still had the fish assemblages within these barren grounds. And so, although overall abundances of many fish might have decreased, the communities were still similar. And so we then asked what habitat characteristics were important in driving any variation in fish assemblages. And so now we're trying to understand how the fish are associating with this new urchin barren habitat type. And so this was a statistical procedure, the BioN procedure, we're using primer E. And of the original 25 habitat variables we noted, this allowed us to identify a subset of those that were really important in associating with fish. And we found 5 habitat variables were the most important ones. These were understory kelp density, the percent cover of understory algae, the percent cover of encrusting algae, the availability of bedrock, and cobble. So right now, the fish, following these changes, appear to be associating with understory things at the bottom. And so that began to let us to believe that now that we've seen that, yeah, the communities have changed, but overall biodiversity has gone down. Some fish species have gone down, but there's still these things there, and they're associating differently with the habitat. And so this led us to ask other questions. And this is something that I do a lot in my lab, is we try to understand ecosystem metabolism, that is ecosystem production. And so we wrote a proposal to NSF to ask what were the effects of widespread kelp loss on ecosystem productivity. And so going back to kind of this story where we go from the kelp forest on the left over to the urchin barrens on the right, the kelp forests have few urchins, loss of fleshy algae, low light levels because there's shading. To transition zones have moderate to high urchins, moderate fleshy algae, moderate light. And the urchin barrens have lots of urchins, few fleshy algae, and really high light because there's no shading going on. And so our hypothesis that we wrote into our NSF was how did the reduction of kelp forests and the increase in urchin barrens impact broad-scale patterns of biodiversity and ecosystem function? In particular, we hypothesized it would result in a decrease in biodiversity and net community production. The biodiversity we saw, there was still biodiversity there, but it changed the players that were there and how they were associating with the ecosystem. But the second part, what happened with net community production, is where we focused one of our sets of cruises up there. And to do this, I worked with an engineer named Mark Tay, who is now retired at San Diego State. And we built these things called SEEP. They're collapsible benthic isolation tents. They're made out of polycarbonate, so they allow full transmission of light, including UV. They're flexible, so as a wave beat happened, they transfer that energy into it, which we verified with fluorescein dye tracers. And so that'll help break down the boundary layers around the kelps for measuring production. They had a big skirt around them, which could be laid on the benthos. We would lay chain on these things, which would create a seal to the bottom. And they had steel framing on the edges, so it held this triangular shape so we can calculate the volume. So now we know the area of the bottom and the volume in here. And these are important for measuring production. And then we would outplant these things to the benthos. And this is one that's placed in an urchin barren. And what you see is inside is a sensor stand that's right inside it, one right outside it. These things had a mini dot oxygen loggers, so we can measure oxygen changes, which gives us an estimate of primary production and of respiration. We had PAR loggers, which measured photosynthetic active radiation. We had temperature loggers inside, and we had pH loggers. And we can measure what's happening right inside that tent that we've isolated versus right outside of it where the ecosystem's in flux and the currents are moving by. And so by isolating these areas and knowing the volume and knowing the area, we can begin to scale up to looking at ecosystem production. So this is what one of these things looks like in the barren grounds. We deployed 3 chambers to replicate them in each habitat, that is the kelp forest, urchin barren, and transition zone, at 10 of the islands for a total of 90 total deployments. Each deployment lasted 36 hours. So we got day and night, and we got a full diurnal cycle. And here's what one of them looked like in a transition zone and what one looks like in a kelp forest. Right here, you can see the sensors inside and the ones right outside. And the way these work is we can take this, and what you're looking at here is the time of day on the x-axis. And I want to thank, I think I saw one of the people on board, a former PhD student of mine, Scott Gabara, who now runs the kelp forest monitoring program at the Channel Islands National Park Service. Scott was instrumental in helping download the data and process this. And what you're looking at is a 36-hour deployment along the x-axis and the oxygen concentration on the y-axis. And you're looking at 3 replicate chambers. And what we see is during the day, we see an increase in oxygen due to photosynthesis. Plants and seaweeds give off oxygen as a product of photosynthesis. But they're also organisms. They have to grow, they have to deal with metabolism, they have to get heal their wounds, they have mitochondria, they all these things. So they respire just like any other organism. And so they suck up oxygen at night. And we see nighttime respiration. Sun comes up the next day, and we see daytime photosynthesis before it stops dropping off again the next day. So we can convert these. We know the time, we know the change in oxygen, and we can convert these to time-dependent estimates of photosynthesis and respiration. And so We did this across the Aleutian Islands. And the first thing we came up with, if we look at net community production, this is just en masse when you balance photosynthesis and respiration together. And we look at that zero mark, which is where the red hatched line is. Above the zero mark, we have net primary production. Below the zero mark, we have net respiration. And right on that zero mark, it balances. This really shocked me, and it really kind of rocked my world when I got these data back. I looked in that there was no effect of the kelp forest loss on net primary production. And so getting back to our hypothesis, the whole reason why I wrote this NSF was that this would result in significant decreases in net community production, and we rejected that. I don't know if there are any graduate students or students on this call right now or young scientists, but one of the things I would say to you is when you go into a study and you have all these ideas of how the world works and you're wrong, don't treat that as a failure. Treat it as an opportunity because this opened up a huge opportunity, which is now some of the stuff dominates the work we do in my lab. And that's because, well, it's not just the kelps, right? There's all the invertebrates. There's the microbes, the fishes, and the other seaweeds, and they're respiring. And so we can look at that nighttime respiration, that is that dark oxygen consumption. And in this, when we bring that in, we see our net community production, which is right on that zero mark here on the, and they're not, it's not, none of them, the habitats are really different from zero. The transition might be a little bit, but really they're hovering right on that zero mark. But if we look at community respiration, which is going negative because respiration is sucking up oxygen, we see the kelp forests have the highest respiration. There's all that huge kelp biomass and all the invertebrates in there that are respiring. And the barren grounds have the lowest respiration. And then we balance community respiration and production, and we look at gross primary production. This is how much oxygen is actually being produced when you take into account for respiration. We actually do see that the kelp forests had the highest gross primary production, and the barren grounds had the least amount. And so this followed, and it's really the story becomes the balance of these two, the respiration and the primary production. And if you look at these, they kind of look like they're mirror images of each other. If I were to shift respiration over to lay it right under the gross primary production, we would see that they begin to cancel each other out, and on net they do. And so really what we found from this is that the ecosystem function loss was not in net community production. It was the difference between primary production and respiration. And if we take that difference between primary production and respiration, the kelp forest lost the greatest amount. The transition in the barren habitats, they lost less, and they're actually pretty similar. The kelp forest lost significantly more than the others. So that's what was lost. It's not net community production. They lost primary production, but they also lost respiration. And the balance of these is what the ecosystem function that was lost. And so we wanted to continue this on looking at the ecosystem metabolism. So we collected all these organisms from the field, brought them back to the ship. We placed them in biological oxygen demand chambers. And here's a sponge in one, here's an urchin in one, here's a kelp in one. And this allows us, by hooking up on the top of this oxygen sensor, a BOD, pro-ODO oxygen sensor, which has a self-stirring bar to mix it up. It's well jacketed, so we can keep them at ambient temperature. And then we can actually, for the seaweeds, we can blast them with any amount of light we wanted. We can measure photosynthesis, respiration of any of the organisms. And we did this with 150 measurements across 26 species. And then the idea is to add these all up across all the organisms in the environment. And then if you put these together and model it back, you can actually begin to understand how ecosystem function lost, where primary production was lost, and where respiration was lost. And so just respiration rate, we can see chitons, gastropods, hydroids, metridium, the white anemone, sea stars, sponges, urchins. We can get estimates of how each of the loss and changes of each of these were contributing to respiration rates. And you can see they all were. And when you start losing them and urchins increase, respiration increases, right? And so the other thing to note is that urchin size varies. It's not just an urchin. And so one thing we can look at and work with another student, Ben Weitzman, this is all these different islands, and these are samples, and these are urchin size frequencies. And we see across the Aleutian archipelago at all these different samples, we began to see that urchin size frequency changes. Some of them were dominated by middle-sized urchins, some of them were dominated by small-sized urchins, and some of them were almost exclusively by small-sized urchins. And so the other way to look at this is if we look at across the Aleutian archipelago, when we look at the percent of the urchin population, very small ones, 1 to 10 centimeter urchins, um, dominated the urchin barrens. There weren't many out in the kelp forests. But as you get bigger and bigger, what you're seeing is the red is shrinking and this teal is expanding, such that by the time you're looking at the biggest urchins, they were really the ones in the kelp forests and not as many in the urchin barrens. So not only did the abundance of the urchins change, but their size frequency changes. And that's important because when you look at the diameter of the urchins and you look at their respiration rates, as urchins get bigger, they respire more. But if you look at scaled urchin ones, so that efficiency, as urchins grow bigger, they respire less per gram. So although a bigger urchin respires more, per gram, it actually becomes more efficient. And so the point of this is that urchin size matters. And so this is a very complex thing to put across millions of urchins that we've counted. If any of you have been affiliated with any of Jim's cruises, you know that any one cruise, you're measuring the test diameter with calipers of hundreds of thousands of urchins every cruise. So it's a huge data set to go to put over. But we do see part of the respiration which changes in the individual organisms. And this brings us to kind of the idea of, all right, well, if net community production was really similar between them, and that it's a balance between respiration and primary production, then we should be able to look at respiration on the y-axis and primary production on the x-axis in all of these chambers. And if the ecosystem is staying in balance, when primary production is low on the x-axis, primary production should be low on the, our respiration should be low on the y-axis. And as primary production increases, so should respiration. And this one-to-one line is a nice theoretical line that says the ecosystem is in balance. Whatever changes are happening, respiration and primary production balance each other out so the ecosystem stays stable. And if we look at all of the deployments, all those 90 deployments we did across that, what we see is in our different habitats, that's actually what happened. In fact, there's no difference in these slopes shown by this interaction of the gross primary production and the habitat interaction. That is, all three ecosystems stayed in balance and they all stayed right on this one-to-one line. So getting back to this idea that, my gosh, we wrote this NSF and we didn't see changes in net community production like we thought we would. Well, you know something? We probably shouldn't, because we needed to take a deeper dive into the metabolism of the system. And what happened with the loss of the sea otters and the increase in urchins and the loss of the kelp forests, the ecosystem metabolism stayed consistent, but the balance stayed consistent, although we did lose that huge gross primary production, but we also lost respiration. So that gave us a really cool way to look into this. And this is something that my lab and Brenda, we published in, I can't remember the journal, PLOS One. The other way of looking at this is the ratio. Here's that one-to-one line. If you're to the left of that one-to-one line, your respiration dominated. If you're to the right of the one-to-one line, your primary production dominated. And yeah, some chambers were a little more productive than they were respiration. Some chambers were a little more respiration than productive. But across all 90 chambers, we see that this ecosystem stayed in balance. And then I looked at this, what's happening in this barren ground? Why do we have such high production out here in this barren ground? Well, we still have clathomorphum. We still have a crusting coralline algae. We have diatoms. And the blue here, those barren grounds are the brightest. And so some of these things are photosynthesizing. We actually saw some of our greatest primary production ratio in areas where you wouldn't expect it, because we weren't looking at these cryptic things we weren't thinking about. So it's a cool way to look at changes in the ecosystem production, biodiversity, all the way down to how the ecosystem metabolism is occurring. So that's a story that we've talked about throughout the Aleutian Islands with sea otters. But that's not the only places that kelp forests have changed. The Eularia fistulosa forest that's clear, but there's also the giant kelp forest here in Southern California, which is where I've spent a lot of my career, from the Monterey Peninsula down to southern Baja. And here's a giant kelp forest, Macrocystis perifera, a nice beautiful shot taken by James Downing. And here's one that you can see that midwater structure. And what happens, it's not a story of sea otters in Southern California, although yes, sea otters historically extended down there. They've been gone, as we know, for a century. It's storms down there. And this is a 1982-1983 El Nino. This is the coast of La Jolla. These are 8-meter waves crashing into houses on the coast. And so it's the waves that cause this, right? And it caused huge pileups of kelp on the beach. And so what happens in Southern California with loss of the giant kelp, work data collected by the Channel Islands National Park Service, which is available, were collected, and Mike Graham published this in 2004 in Ecosystems. We're looking at an interactive web. We're along the bottom. We're looking at primary producers, our kelp, other macroalgae, our coral and algae, which I just showed you is important for the Aleutians, phytoplankton, phytodetritus rolling around. And these things are feeding up into primary grazers here. And those grazers feed up into predators and then tertiary predators. And so these interactive webs are a nice way to utilize the complexity of the system. But what if you just take the kelp, leave other macroalgae and crush those corallins, the phytoplankton there like we saw in the Aleutians, and just take the kelp out? Well, if you just take the kelp out, we see a 37% reduction in species and a simplification in the food webs and a loss of tertiary predators. And so we see similar changes in biodiversity and interactive webs in Southern California as we saw in the Aleutians. And we can actually go in and say, all right, well, let's take a look at, we haven't done the chamber work here. We're about getting ready to do some of that work, but we can collect water samples and ask how much CO2 is in the water. That is, how much, photosynthesis sucks up the CO2, respiration gives it off. We can look inside the forests in blue. We can look outside the forest, or there's no forest in orange, kind of getting back to that color scheme we saw in the Aleutians. And this interesting thing is at the surface where it's bright and in the midwater, we see a drawdown of CO2. The blue is lower than the orange. That photosynthesis is drawing down CO2. But at the bottom, where it's darker, there's all those benthic invertebrates, there's the benthic microbes. We actually see an increase in CO2 relative to outside of it. So we see more net community production, CO2 uptake in the bottom and at the surface and the midwater, but we see more respiration, CO2 production at the bottom. And so this is another one. Kind of the last story I'll end on mine, and that this is bull kelp, Nearshore here, and we looked at this, and I looked at this up in the Gulf of Alaska. This is work I'd love to do the Pacific Northwest. And what we've had happen, and many of you on this call are probably very aware, we've had maybe up to a 90% reduction in bull kelp along the Northern California and Oregon coast. Started with a marine heat wave and a strong ENSO around 2014. It's propagated through a change in feeding behavior in the sea urchins. They've now emerged, and they're grazing everything down, and they're not allowing the kelps to recover. And so work to look at Nearshore. This is Kachemak Bay, the Kitsitna Bay Marine Lab. Some of you may have been there. This is before that NOAA rebuilt it into a nice, huge thing. But we worked up there, and Kitsitna Bay is a beautiful place to work. And we did the same kind of deployments for our water collections there, and we looked at CO2 at the surface and at the bottom, and we see the same thing. At the surface water, bull kelp draws down the CO2, and at the bottom, we see production of CO2. So you can't even say that when you lose the forests, do we actually lose photosynthesis and CO2 drawdown? Because now it depends on what depth you're at. We have to integrate across the entire depth profile. So this becomes a very complicated story, but it's one that just with time and methodical going through, we think we can address. So, and these are the kinds of things that I would love to, when I'm up in Oregon, that I want to continue. I'd love to work with the Oregon Kelp Alliance and with your organization there. So anyway, so I'm going to go ahead and end this because I think my time is coming coming up. Um, so although I was giving a chapter that was primarily Jane Watson, Brenda Kohner, and I, which covers everything from Alaska down through Mexico, um, the actual field work on this, really I want to send my thanks to the Kohner lab and my lab, the people that joined us up there. We've got a museum, a traveling museum called Underwater Forests of the Aleutian, which documents many of these. Um, Great to have a number of graduate students working with us. We've also, I want to thank a couple of collaborators, Melissa Good, Ju Young Kim, and Mandy Lindberg, who contributed parts of this. Ju Young Kim in particular, bringing the metabolism aspect to this. And then NSF and our funders, and then the crew. This is the crew of the Oceanus, but also the Tommy Thompson and the Point Sur. So from this, I think I'm going to stop sharing and get back. And so I want to thank you for hearing a story about kind of the loss of kelp forests and ecosystem function and diversity. Um, and I'll happy to answer any questions.
Thanks, Matt. And we're also very excited to have you come to Oregon soon and continue your kelp forest work.
Mhm.
Um, Also, just want to say, love the Darwin quote. I often compare kelp forests to our terrestrial forests in presentations because so many people understand how beautiful it is to walk through a forest in Oregon, especially.
Mhm.
Okay, so if you have any questions for Matt, please enter it in the Q&A feature. Christine entered one, but you may have answered it, but I'm just going to ask you anyway because we've got
Okay.
some time. So are you saying that the idea of keystone species with sea otters has kind of been nulled out by your study, or is the main point of that story that kelp forest is being depleted?
I don't think it's at all being nulled out. If you take, the concept of keystone species has um, had these debates. And I like the symposium that came out of the working group in Hawaii, where they kind of nailed it down. And so the idea of the keystone species is right that it has a disproportionate effect on its ecosystem than its abundance. And so sea otters are certainly in low abundance, and it's clear from what we've seen on these talks, and you can see more today, and from the way I set this up, that it's clear that when sea otters were extirpated, the ecosystem changed. It didn't take millions and millions and millions out, right? Sea urchins, you would have to call tens of millions, hundreds of millions to get some sort of a change back, right? But if reintroduction of a few sea otters may help bring some of these things back, right? Now can be controversial, and whether they would eat the sea urchins with the gonad condition. So, rambling answer, no, I don't think it nullifies it all. I think the concept of keystone species is just as important now, and it's just as true now. I think it's gotten lost in the dialogue over, and a lot of things have been called keystone species that maybe don't quite fit that bill, but I do think that sea otters are it. In Southern California, where we don't have them, no, I think it becomes storms and productivity. There's some interesting debates particularly between Mike Foster and Dave Shield with Jim Estes about the applying this concept to Southern California. Those are salient arguments to make because I don't think the otters are a keystone species in Southern California. They don't occur there, but I think bottom-up forcing, storms, productivity, temperature, those things become more important in Southern California.
Great, thank you. Susan has a question. Do your measurements of CO2 take into account increases due to global atmospheric CO2?
Um, yes, um, but over the time scale at which we're doing them now, if I had done these same measurements 20 years ago, and I did them in 20, I do hope to do them in 20 years. We have measured these things all through British Columbia as well. And we've measured these things off of the northern Washington coast around Neah Bay, Cape Flattery area. The answer to that question, I'm gonna have to say, how do these systems look in 20 years from now? Because you have from the atmospheric CO2 coming in from the surface, but right now I think just on the ground measurements, the CO2 being produced by benthic respiration is big. And there's a lot going on the benthos of a kelp bed and an urchin barren. And so the whole system will probably come up, but these relative differences, I don't expect them to change. Um, we're certainly not near saturation of CO2 in seawater. We can get these things, CO2 up to 20,000 microatmospheres, where you can get the pH down to 5.1, which we do in my lab when we're doing certain experiments. When we're doing laboratory experiments, just take a pure CO2 can, give it 2 second blast into a thing of water, and that pH goes from 7.8, 8.0 down to 5.1. And so we can put a lot more CO2 into the water. So I don't think this is as a climate change study. We've been doing a lot of that, but the time scale over this, I need decades to do this, and I do hope to do that.
Thank you. All right, we have another question from Alexa. Is there any way to reintroduce kelp to an area where there's been deforestation, and would it be helpful?
Yes, there is. So we do reforestation in my lab right now. We're working on Catalina Island, where kelp was lost due to the same marine heat wave, the blob, if you will, in 2014. But instead of being a story about sea otters, an invasive species came in, Sargassum horneri. It's now along the whole west coast from Central California down through Mexico. And it's a warm water tolerant species, and it has a microbial component, probably a vibrio, that's associated with it that is helping keep kelp excluded. We've been doing some work removing the invasive species, and kelp is recovering on our plots in Catalina. We're getting really good recovery. Um, and my wife is a restoration ecologist on land, and invasive species control is one of the things in grasslands. Get rid of them, and things can return. So, yes, to get rid of, reforestation, I've done some talks on this with agencies. First thing we need to do is you've got to figure out the stressor. Why did the kelps, you can't just plant kelp, you can plant kelps, but they're going to disappear. They disappeared for a reason. They'll probably disappear again. You've got to get rid of that stressor. If that stressor is urchin grazing, got to get rid of the urchins. Now, that's what they're doing in Central California, where I've been working with UC Davis, Sonoma State, and a little bit with the University of Washington, trying to get commercial fishermen, dive groups to remove sea urchins. It's a monumental task. You're talking probably billions. Just probably can't be done at the scale at which we can work. Disease might take them out. That's my thought is, at some point, urchins are famous for diseases, especially in warmer water. Disease might take them out. But as far as you can plant, you can transport their sporogenic material to sites. They will naturally seed, and they can recover if you remove that stressor. Climate change is one we can't deal with. Now, kelps are disappearing in certain areas due to climate change, particularly in their lower latitudinal limits. Um, Maybe work being done in a lot of places is trying to find warm temperature strains, tolerant strains of kelp by doing simple garden experiments and outplanting warm tolerant strains. So it's not hopeless. You can transplant them, but not until you figured out why they disappeared in the first place and remediating for that. Invasive species, sea otters, urchins, temperature. If we can remediate for that, kelp reforestation is not, it's totally possible. I believe that.
Excellent. Do we have any more last minute questions that just popped into your brain? I'll wait and see. I have a question.
Yeah.
Where where would be like the warmest part of the world that we have kelp?
The warmest part of the world. So now you've got to, you've kelp right as a catch-all for an order of dozens and dozens of species. And some of them are much more tolerant. Work, there's a paper that came out by Mike Graham and colleagues that followed up some work by the people at the Darwin Station in the Galapagos, Kathy Ann Miller, who's at the Berkeley Aquarium, where you have kelps on the equator. Now there's equatorial upwelling, so the water's not quite as cold as you might think. Along the coast of Peru at 6 degrees, you have the Peruvian current, you have strong upwelling centers. So we have kelps there, but the warmest part of the world, if I had to take a stab at it, I would say the south coast of Australia, which is in trouble. Giant kelps disappeared, but they have a clonia there, Clonia radiata. And so if you want to go look for kelps that are dealing with a warming world, it's probably, you go talk to Thomas Warnberg, um, and start looking at the southern coast of Australia. That's where I would probably target looking where the answer for that one is.
Great. Okay, well, I think we have answered all the, oh gosh, right in the nick of time, Eric. All right. Do you think sea star recovery outplanting could be a viable strategy for bringing back kelp or combating urchin barrens?
Certainly is a topic right now. Jason Hoden at in Washington, we're working with him to rear these things. There's talk about trying to reintroduce sunflower stars, in particular, Pycnopodia. I mean, it's something that's that voracious of a predator. But yes, so if I were to say predator mediated, right? Predators do one thing is they forage 24 hours a day. They do rest, but it's 24 hours a day over the whole scape of landscape. People, we can get to areas we can take a boat or we can dive from shore. So we can't touch huge areas of the coastline. So it's not going to be people culling the urchins, maybe in small areas, but predators, right? Predation is going on constantly over huge areas. So sea star reintroductions, absolutely, I think it's a possibility. Um, Problem, a lot of the urchins are in poor quality. Their gonads have been reabsorbed. They are not as energetic. So some, you work out of Brenda Koner's lab has shown that the otters will avoid poor quality urchins. See whether sea stars do or not, I don't know. So yes, I do think there's some legs in that. I do think it is worth investigating whether the reintroduction of sea stars into the areas, particularly Pycnopodia, laboratory rearing them and then releasing them. There's whole things about, laboratory rearing anything and releasing it. We've got to be careful of the dangers that are posed there. So it's a cautionary tale as well, right? Because we can do more harm than good at times. But I
Mhm.
do think there's potential there.